So we were discussing about nucleic acid-based therapeutic drug delivery system in the previous class. We have seen the role of liposomes in that particular delivery of gene therapy. When in addition to the previous in the targeted drug delivery, we have seen different delivery systems in the nanotechnology, nano level, and nano-sized level. So in that we have got a different approaches where we are going to enter into the nucleus, because that is very much critical in the gene therapy. And we have already discussed about the advantages of liposome when compared to remaining dosage form, why it is being critically taken, why this product is entered into the market. Now, vaccines and some of the drugs are also made in the form of liposomal delivery systems because of these advantages. So different types of liposomes we are going to do depending upon requirements. If you see this slide, we are going to have a stimulus based, and depending upon the type of ligand, we are going to have a different approaches to incorporate in the liposomes, so that we are going to utilize them for the efficacy and the safety. These are all the stabilizers it is being listed, so that we need to consider them any drug targeting and liposomal delivery system or any gene therapy where we are need to stabilize the drug delivery system. Most of the times we are also seeing what are the target diseases. Most of the times there are so many number of diseases, why we are concentrating about the gene therapy in particular only. And in nearby future, we may be having so much of importance of this, and much widely it will be used. But we have recognized most of the times in the genetic therapy, in the genetic disorder for the particular person, or it may be due to heredity. So the concept of gene therapy to treat the individual diseases and diseases caused by the heredity. So the steps involved in that we have already seen that. So first we are identifying the defect gene and cloning it outside and making a healthy gene and which is not having any defect, and identification of the target cell or a tissue organ where we need to insert this corrected gene, and insertion of the normal functional gene into the posterior. This is what it is being done. So in our part in that particular case is how to deliver it. So the identification of the defect gene, it will be known by the type of the symptoms. So from the gene database also we can predict in the in silico models which are present what are the particular segment which is going to cause. And if it is matching with the actual gene sequencing, whatever it is being present by the individual of a patient, so then we need to take that particular segment outside and we need to correct them by some of the gene therapy techniques. After that, we need to take where to take this, and we need to select it and insert this corrected gene into that, so it will give the solution to the problem. This is what we have already previously discussed. So the types of gene therapy. We have come into there. Now we will be having a question: what are the different types of the major types of the gene therapy? This is what we are going to see now. There are two types of the gene therapy. Here we are using genetic material as our drug without giving any drug. It is actually what we are going in this particular case is we are correcting at the molecular level. So in the correction at the molecular level, we need to have therapy. What way we need to do it based on that sequences of what is going to happen and what cell we need to attack in the gene therapy. Based on that, it is divided into the two types. One is somatic gene therapy, another one is germline gene therapy. So what is the somatic gene therapy? What is germline gene therapy? Where we need to use in our diseases generally which is present in the population. I have already mentioned that don't think that these are all the genetic defects are more prevalent. It is not like that. They are the rare diseases, and we are considering as a orphan category, and very less population will be present in the range of 0.1%, less than that only it will be present. But we cannot wait till the death, whatever the defect they are having, or they are losing their life in advance before they need to have completion of their task and the purpose in the life. So for that purpose, we need to do something. Even though it is a smallest population, we need to consider them, even though the expenses are very high. So this is being taken into the account. So there are two types of therapy. One is the somatic, and the other one is the germline. So in the somatic gene therapy, the therapeutic genes are transferred to the somatic cells. That is the reason we are cutting a somatic gene therapy. The somatic cells are the non-sex cells. It is not having a like spermatozoa, or it is not like ovum like that. It is actually somatic cells, non-sex cells. So most of the times it is in the mitosis. They are involving, and in the body of the patient, generally the target sites will be bone marrow and skin cells. Sometimes it will be a nerve cell. Sometimes it will be a blood or particular tissues which is accessible from the outside and near to the skin. So it may be a certain part in the breast. Like that, it will be having, it is non-sex cells which will be targeted in the somatic gene therapy. Whereas the germline therapy, the therapeutic agents are transferred into germ cells, that is eggs or sperms, or it is having. So this is actually this is to be done in a view that the disease will not be inherited to the later generations, because when we are correcting at the egg and the sperm level, so where the defect is existing, so it may be a maternal or paternal defect. In the maternal defect, we are going to consider the egg first to treat the disease. In the paternal one, we are going to take the sperm cells into the account, and it will be corrected in advance. Sometimes we know that we are going to have things which are going to have is maybe encountering the changes at the zygote level. That can also be done. But the main aim is the disease will not be inherited to the latter generations. Otherwise, it will be going in a generations together. Sometimes it may be some generation may be carrier. Some generations may be affect us. So this may be alternatively it is present, we don't know. So, but it is getting carried. So we not to avoid it. It is good that we are going to attack at the molecular level in the germ cells itself. It is inheritable and passed on to the latter generations. That is the reason we are considering this type of the therapy. It is very dangerous, and it is very common. Even a small defect in another place in the gene, either it is in the somatic or in this germline, it will cause a lethal effects, and it may make the function of other organ gone, or it may be altering characteristic and properties or the mentality of the individual also. So that is actually affecting physical and mental and characteristics of the patient. That is the reason this gene therapy is considered to be very much taken into the account. There are ethical grounds are also present, because we are correcting the genes at the egg and sperm levels, and nowadays it is being appearing that why can't I give a birth to the offspring, my son or a daughter. They are having like maybe good sprinters. They are very good in run, and they have got a great height and physique, maybe get a six-pack like that, and maybe having highest IQ, maybe having a serene-like traits, which are having like saint-like traits, blended with highly intelligent, with resistance to the great diseases. Such offspring, why can't I make it? So these are all the explorations which will be done if we are correcting and doing changes in the molecular level at the germ, in the germ cells, incorporating the genes which are responsible for these traits. There is an idea that we can create an individual is a complete package with respect to mental and physical and intellectual traits. So this is actually what will happen. Later onwards, it will inherit, and it will be passed on to the next generations also. And it will come into an idea, so it will cause us a greater ethical grounds are required to be considered. Because if we don't know the correct way of doing it, if anything which is alteration or misuses of technology, it may result into a different types of characteristics or traits, which is difficult to humankind. It may be the problem. That is the reason Even though the advances are present, they made the bar not to do such things at the germ level. Only it is required to be restricted to the disease treatment only, not to enhance the normal one individual to the supernatural individual. The natural human being has got some certain limitations with respect to his physical and mental and intellectual traits. So we don't want to have that is maintained, but we want to make it further advanced. In that particular case, we need to consider, so they kept the bar. But the disease treatment and doing the things in certain primitive animals for the explorative research, if it is going to give you a better insights into the medical, that is the pharmaceutical and medical industry to treat the diseases, it is good. Most of the time, genetically treated stem cells, when reintroduced into the patient's body, are expected to be naturally travel through the bloodstream to the bone marrow, and genetically changes in the reproductive cell or embryo before the stage of differentiation, in fact, it is a single zygote. So, would affect all the future offspring that particular person. This is actually what the logic is. It means some person is having an inheritance of a night blindness, and all the people in that particular generation is born with night blindness, or that may be a color blindness, somebody may be having a hemophilia. So such problems are inherited with a small segments in the gene, that is a small changes in the base pairs that has caused in certain level of their ancestors because of the mutation. So that the entire generation is getting affected with that particular problem. So why can't we make by having a defect changed in the reproductive cell itself, so that the next offspring will not have that particular problem. This way it is okay, it is ethical, and we are having a treatment, but not that advancement. In the name of advancement, we are doing unethical means of doing gene changes and making a most of the times we have got a transgenic animal, transgenic plants. So they are having a hybrid characteristics, traits, which we are seeing in the agriculture. So they are having a size, tomato is having a size of coconut, and we are having a seedless mangoes, where the seed is not present, just like your grape. So these are all the things which is appearing that it is having certain things like vegetarian chicken, maybe cell grown, which is having in the cell lines in the biotechnological laboratories. So which is having the same nutrients and the taste and texture and feeling same as that of the regular one. So this is actually what is killing of so many animals, and methane emission and the gases of greenhouse effect, they will be reduced, such things which are there. So at the plant level and the animal level, it is being keep on going on the research. But in the aspect of increasing the traits of human beings to have extra caliber and the skill, so it is generally got restricted. It should be restricted also. Now coming to the two somatic gene therapy, that is actually a correction of the defect at a particular tissue, at a particular organ, and it will not affect the next generation. It is particular the patient only, not to the next generation. So this is actually what the somatic gene therapy. In that we have got one is ex-vivo gene therapy and in-vivo gene therapy. So what is ex-vivo gene therapy? This is actually what ex-vivo means. It is live only. We are not going to kill. It is not in vitro. In vitro means we are going to use any non-living organisms and non-living things where we are going to handle, and we are going to have a dissolution test. There we don't have any tissues, we don't have anything. We have got a non-bio, which is having no life in the dissolution test apparatus and the tablet and the medium. So just we will do it is in vitro without having any life and outside our body. Whereas in vivo means within the body. We are going to bioavailability studies. We are going to give into the body, and we are calling it as a bio in vivo. So generally, I made it. We need to write it in the most of the times such things which we need to write in the italics. So this we need to consider generally. So we are having the ex-vivo gene therapy. It is a middle of these in vivo and in vitro. What we do in this particular case, isolate the cells from the genetic defect from the patient. We take that. It is actually life it is having, but we are growing outside. That is the reason it is called as ex-vivo. It is not within the body. To grow the cells in the culture, it is outside in the laboratory. And select the genetically correct cells. We have got a treatment, and some cells are present in the original defect, some cells are corrected. And we collect the correct cells, and again we tend to make them grow and transplant the modified cells into the patient. This is actually called as ex-vivo gene therapy. It means we are doing certain interventions outside to our body, and again we are doing that entering into the again body to treat the disease. This is called as a ex-vivo treatment. This is just like your test tube baby. Why you are having this artificial fertilization? Because there is a defect in the uterus or environment of the progress of the spermatozoa, and not having enough spermatozoic cells to reach the follicle, so fallopian tubes. In the particular case, we are making in vitro fertilization to have, that is an ex-vivo intervention, and again introducing the zygote into the mother. In the surrogacy, we are introducing into the different mother. In the case of mother is having a defect in lack of uterus and not having a proper place or expansion or defect in the uterus, so then surrogacy will come. But in all the cases, there is an ex-vivo intervention is there. In the case of artificial insemination and making test tube babies, in that there is not a treat, it is actually one type of making an intervention. We are not going to do any changes there. Just we take it and add it and do it. But in the therapy, we make some changes in the genetic material itself. We need to do some changes. Don't get confused with the ex-vivo is not a gene therapy. We don't call it. It is a in vitro fertilization where we are having a fertilization only in vitro, and that is the ex-vivo concept, and making it to entering it. Here we are not doing any therapeutical intervention. Just we are doing a joining and making them to form a zygote outside, because the conditions are not available inside. But here in the case of gene therapy, what we do, we genetically correct the cells. We don't leave like that only. We correct the defected genes, and we are making it to entry by the transplanting them, where we have got a interventions of our pharmacists which are present. The second one is in-vivo gene therapy. So in the somatic gene therapy, one is in-vivo. It is not having any everything is going on within the body only. So the direct delivery of the therapeutic gene into the target cell, into the patient's body. This is actually one way, and carried out, it can be carried by the again two types, that is viral and non-viral vectors. Now we are entering, and little bit concept is more and more clear now. So we have first divided into the gene therapy into two types. One is the somatic, another one is germline. In the somatic, we have got again two types, an ex-vivo and in-vivo. In-vivo, we have got again two types. One is viral and non-viral vectors. So we are categorizing our gene therapy with a broader sense. So if you have been asked the question, to classify the gene therapy, you need to make a classification chart. So by doing like that, you will be having a good marks. So you categorize, you take a piece of paper and arrange like that, and you write that classification only, instead of writing all this matter. If you've been asked what are the different types of the gene therapy approaches. In the in-vivo gene therapy, we are going to do by the viral vector and non-viral vector. In the non-viral vector, other than the virus we are going to use, there we are going to have that particular delivery possible by the non-viral vector. So in-vivo gene transfer is necessary when cultured cell cannot be reimplanted in the patients effectively. Why we are going to adopt this, even though it is a little bit complicated? Why we want to do it? So the outside cell we are not able to introduce again into the patient effectively. That is the reason we are doing an in-vivo intervention that we need to consider. If there is an ex-vivo intervention is there and it is then, it is good that if any defect is there, we will throw it out. Exterior only, we will not make. So this is actually not possible to reintroduce and implant the successful gene and the corrected gene into the patient. Then only we can be using it. We are not able to precisely make that possible, and we are doing this in-vivo gene therapy within the body itself, because the problems of without using it. So in the vector gene therapy, now we are having one is the transfer of the desired gene into the target cell. A carrier is required. So the vehicles of the gene therapy are also known as vector. What are the vehicles? Are the carriers? So that is called as the vector. So there are two main classes of the vectors. One is a viral vector, another one is a non-viral vector. That's what we have already previously seen. And now we are coming to the gene therapy, where we are going to use the viral vectors. Virus is a great thing, even though it is having a harmful effect sometimes, but they are going to be helpful so many times. And there are so many useful viruses which are making plants sustaining their lives, and universal survival is, we can say it because of these viruses only. But there are certain mutant viruses which is causing a disease, it is giving a problem. So there, where there is, if it is good is prevailing in the society everywhere, there will not be any harm. But there will be a changes and alterations and mutations that will be made by the environment. So when we are spoiling the environment, so the mutations will occur. The mutations will go out of hand. That will cause the damage. So here we are taking the viruses for the good cause. So the viruses which are causing the diseases, and these viruses are going to help us as a carriers. Just they are instrumental enough to do our duty on our behalf. So the viruses introduce their genetic material, because they are having major part is genetic material. If you see the coronaviruses, it is having a internal genetic material and the spike protein, outside membrane, and the spike protein. So that much simple, it is present, and it is causing a lot of damage to the 60 kg body weight human being as an average, and making him to die also by taking a reign over the cells of the body. So these viruses are they more prone to get introduced at the nuclear level, at the molecular level. So we are using a molecular pharmaceutics. We want to deliver the drugs to the molecular level. At the molecular level, to the near to the nucleus, even in the nucleus, we want to make a changes in the particular sequence. So in that particular case, these viruses have got natural tendency to enter that particular site. Why can't we use them? So this concept has come, and the viruses introduce their genetic material into the host cell as a part of the replication cycle. And what we do for that particular purpose is remove the viral DNA and use the virus as a vehicle to deliver our therapeutic DNA. It means now, whenever we are taking a viral vector, the virus will not carry their own DNA. In the place of it, we are going to have our whatever the therapeutic DNA we are going to use. This concept has made a lot of changes in the production of your vaccines. It may be a production of your human insulin. So many changes which could be made by doing this removal of the viral DNA, placing a character DNA to have a particular gene that will produce, or it will send its characters to the patient. So such things are present. Now, in the again viral vector, there are 4 types of the vectors are present. One is retrovirus, another one is adenovirus, and the third one is adeno-associated virus, fourth one is herpes simplex virus. So these 4 viruses are there. Don't get confused. We are going one by one and going a little bit deeper. And now you focus on this particular viral vector, and we have got this 4 viral vector. Now the question comes, sir, where we need to use what type of virus vector? So where we need to use, depending upon our target site, we need to select any one of these 4. So what these are having a differences, and how they are making it attacking to the particular cell, and what type of the cell they have got a specificity. So we need to know first. Then we need to select any one of these viral vector if you want to use a virus as a delivery system without using any nanoparticle or anything. Then we can use it in the gene therapy. I hope you have understood till now the concept. So this is actually even at the B farm level also you might have already learned. So we are going to see a little bit deeper. Now we will be seeing it, and only the thing is we need to select them where we need to use it in our correction to the patient disease. So the first one in the particular case is retrovirus based vectors. So in the retrovirus based vectors, the retroviral vector binding to the host cell surface, that is patient cells, that is normal host, we call them, and then it will uncoat of the virus will be taking place after. So this is actually this uncoating phenomena. Most of the times the binding which has got a specificity. Most of the it is having a specificity. We have thought that the retrovirus binding at the host cell surface. The host cell surface has got a particular specificity. So this is actually what it is being done in most of the cases in anti-COVID drugs also. If we are using some antiviral drugs, the specificity to attack or attach of this spike protein onto the cell surface will be lost. So they will not be getting adhered onto the cell surface of a patient. Then there is no question of entry. So once they are adhered only, uncoating of virus will be taking place and release of the vector viral RNA is present, that is entered, and DNA production and the replication. They introduce the RNA, whatever the release of the RNA, they will get introduced into the DNA of that particular cell. And now the DNA is not the original DNA of what the patient is actually having. Now it will be taking the introduced RNA portion is also introduced there. That is going to ride and guide the duty of that one, and it will be having a replication. So the integration of the vector into the host genome of the dividing cell and expression of the heterogeneous gene. So most of the times what will happen, it will be used most of the time, this type of things will be used in the ex-vivo gene therapy. What we do, we take the external cell, host cell outside, and we introduce the genetic material of the RNA into that DNA and correct that particular DNA. We already see now we are going to have a correcting it. Then we will be replicating that particular cell, and after that we are introducing it. So this is used, this type of retrovirus is based on the usage of in the ex-vivo therapy, and it is used to correct your say floating tissue like your blood, or it is you want to introduce any proteins, you want to make it ex-vivo. In the case of bioreactors, you want to make a proteins, that is the antibodies which you want to generate it. So what you are doing it, we are making a viral RNA which is replicating very faster. That is the reason we are taking that particular segment and introducing the DNA of what we are going to have make a particular antibody, and we are joining them and we are introducing the same into the bacteria. The bacteria cell now it is lost their original DNA, and they are using this particular DNA and synthesize the drug, that is antibody, on our behalf, and we are going to have the same one which we are going to have so much of human specific, and it is specific to the human that is being used. So this is actually a retrovirus based vectors are very much being used in so much of you are going to use a recombinant DNA technology, or it is antibody production, or it is synthesis of the so many proteins, and it is in the usage in the insulin. So these are all vectors which are used to introduce the material of a desired one into the site. These viruses are being used. The second one is the adenovirus based vector. That is actually the adeno-based virus vector enters the host cell in the acidic environment, and the protease in the virion, so they get activated and proteolysis of the virion will be taking place, while the DNA is released into the cytoplasm, and DNA is transported into the nucleus, and it is going to do serve the purpose. In particular case, the genetic material is a double stranded. In the previous case, retrovirus, we have got a single stranded. That is RNA we are having. Here, it is a viral vector is DNA based. So this viral vector is a DNA based. So you know that I don't want to make it further going little bit deeper in this. I want to make it that is revised, because you know it. So the difference between the DNA and the RNA, one difference you know that one is RNA is a single stranded, DNA is a double stranded, and DNA is having a deoxyribose as a your pentose, which is present in the genetic material, whereas RNA is having a ribose, not the deoxy. It is the oxygen is still present. It is not the deoxy. So this is actually what are the differences in this particular two virus vectors in the genetic material. So we are going to have this advantages. So to building upon our requirement, we need to use them. It is a DNA-based virus, RNA-based virus. One change is single-stranded and double-stranded. The second change is the ribose, which is present deoxyribose. Oxygen is not removed, deoxyribose. That in the fructose sugar, oxygen is not present, but in the RNA, it is oxygen is present. Now you may get the question, so what is the advantage of having this DNA and RNA? Most of the times, RNA is more prone to get degraded because of the presence of oxygen, and it is a single-stranded. Their life is very less. Whereas the DNA is a little bit more stable when compared to RNA, and deoxyribose, which is present, it is having a longevity. That is the reason our genetic material is present with DNA based, and the DNA being used as a template to synthesize the RNA, so that our original traits will be kept as such without any changes. It means my every cell has got the same DNA that got protected, because it is also got protected with the shield of these phosphate groups and pentose, and the basic pairs are present, already got a double-stranded, and they have got a spiral, they have got folded, everywhere they have got a protective mechanism. Because of that reason, that original template will be protected, and the small segment in that particular DNA, it will synthesize the RNA. That RNA will be present, and depending upon a requirement, that will be triggered, and it will be going into three types. One is ribosomal RNA, rRNA, and messenger RNA, that is mRNA, and transfer RNA, that is a tRNA. So these three are going to be present, and they are having a not like permanency. So they are not having a permanent role. Their role is present, and they will be getting destroyed, not like a DNA, which is present, which is in the every cell. So the ribosomal RNA and the messenger RNA and transfer RNA, it will be varying with one cell to another cell depending upon its function. Say, for example, my body want to make a ear. Its shape is something different. Its shape is different. The proteins required for making it, the color of the eyes, it is either it is gray or brown. It will be completely different. Complexion is different. So we need to have a changes in that particular region or purpose. We want to make it. There, these mRNA, messenger RNA, and tRNA, it will be keep on altering it. So that is very much flexible. So that part is present. Once that part is made, and say, for example, we have got eyes. The eyes, what the birth we have taken, after that, there is no further enlargement, or they release, or they decrease. Whatever it is present at the birth, it will be taken till the death. But whereas my ears, it is keep on growing. So why this, when I am child, my ears are small and I am getting bigger. After the particular period of the time, the growth will be stopped. So how it is being happened there? So my complexion and stoutness and fattiness, I'm little bit keep on growing. So this is when it is growing. Some parts like the eye, they are not growing. If you are thinking that when my eye is present in the child, the size of eyeball is present, if it is keep on growing, so my skull have to break because of this pressure of the growth of this. It is not growing. The exterior ear is, of course, it is growing. The exterior nose, it is becoming little bit bigger when compared to where my childhood, and it is being bigger and bigger when the person is growing further. So it means that there is some traits in this RNA, it is making a differentiation and working at different sites, and they will be getting functioning and got destroyed. It will not be present permanently. Once that purpose is over, they are getting destroyed. It means this is a trait we need to understand. This you know it. So this RNA is little bit appearing as a temporary, whereas the DNA is appearing as a little bit permanent in nature, and longevity is in nature. So this protein viral and viral DNA is released, entering into the cytoplasm. DNA is transported into the nucleus, and it will start doing its purpose. So most of the times you know it, DNA will not serve its purpose directly. It want to purpose anything for function. Again, DNA generates a single-stranded RNA. The RNA is again taking the help of messenger RNA and your transfer RNA, and you're taking amino acids, and it is converting into a protein. That protein is actually helping us. It means a triggering will be present to a DNA, and it is resulting into the liberation of RNA. In the trigger is present, it may be a depletion or present. So, or any requirement is there from the external stimulus. So this is actually what is making an alteration in the DNA to release the RNA to synthesize. This is what we are doing in the vaccines. We are doing in the vaccination, whether it is Covaxin or Covishield, when you are doing it, so we are making some trigger in a particular gene in such a way that RNA is released to synthesize the antibody, so that the body will be getting safe by making these proteins, antibodies, which are required to be present in advance, because there is a trigger we are giving it. That environment is producing a trigger that is resulting into the synthesis of the proteins. The protein antibodies are helping us. It means this trigger is coming from the external world. So previous case of trigger of stopping your eyeball expansion and synthesis is given by the internal world. Internal world means we are giving by birth itself the genetic material has got this programmed way of doing it. So my eyeball will not be going when I have taken a birth. My eyeball is the same. It may get shinked, it may be getting glaucoma or some defect will come, but it will not grow. This is actually what the basic function of this nature. It has got some authority to control by at a particular period of time by the internal trigger or external trigger. So this is the one which is making wide differences in the vectors also. And primitive animals like even viruses is the least primitive living organ. There also these differences are present. That is making the virus easy to spread and maintaining their life longer depending upon the traits what they are having it. We need to understand, because we want to have a invention of a medicine or a formulation for these vectors which they are harmful. Another one is adeno-associated virus vector. Adeno-associated virus vector. It is a human virus that it can integrate into the chromosome, and it is a single-stranded and non-pathogenic small DNA virus. Now you may get the question, sir, you already mentioned double-stranded is present in the DNA. Now you are calling it as a single-stranded DNA. So when you are calling it when it is a double-stranded DNA, it is a single-stranded DNA, you should call it as an RNA virus. I think you may be having that mind. So if anybody is having a answer for this, why this single-stranded non-pathogenic small is called as a DNA virus, why it is not called as RNA virus? If anybody is having a small clarification, they can unmute themselves and say, why can't we call this as a RNA virus? It is a single-stranded RNA virus. Do you have any idea? Most of the times, RNA is a single-stranded, DNA is a double-stranded. That will be existing. When it is a single-stranded, then it should be called as a RNA only. Then why it is called as a DNA? DNA is a template for the RNA, and when it is a single-stranded, it is appearing as RNA. So why it is calling it a single-stranded DNA virus? That is the question. Why you call? Okay. So if you remember it, we have already mentioned a little bit 2 minutes back. In the DNA, we have got deoxyribose. That is actually making its function to maintain longer or not. Only the complementary strand automatically it can come. So the deoxyribose, which is present, there is no oxygen in the ribose pentose moiety, that is making a DNA. In this particular virus, the deoxyribose is present, not the ribose as such. That is the reason we can't call it as RNA. So that RNA is less stable when compared to this. So adeno-associated virus vector, we are having a deoxyribose only, but it is present as a single-stranded. So this is actually what the virus vector you need to understand. So adeno-associated virus, that is AAA, enters the host cell, becomes the double-stranded and gets integrated into the chromosome, and it will serve the purpose. And the D1 is herpes simplex virus vector. Herpes simplex virus vector. So virus which have a natural tendency to infect a particular type of the cell. They infect and persist in the nerve cells. Specifically, it is in the nerve cells. Most of the times, the herpes simplex, what we are seeing in exterior is herpes zoster or anywhere. the nerve cells are getting affected. Now we know that these one which can be differentiatedly used it for our purpose. This is a simple technique if you are seeing it. So there is a problem and you want to have your nerve cells are getting regenerated. Say one, I would like to say a Parkinson disease. So this is actually you got a problem with the nerve cells because of the degeneration of the nerve cells is happening. You want to regenerate it. So you want to introduce the gene material in the nerve cells. Then what is the viral vector? It is better for you, herpes simplex, because it is viral specific to the nervous system. So retrovirus, like a blood, which is a specific one, which is good, that is actually retrovirus. If you have got introducing of a gene defects, then adenovirus one is the one is good. If you see here, each one it has got a specificity and making them. In the adenovirus, what is doing? It is a single-stranded adenovirus virus enters into the host cell, become a double-stranded and gets converted into the chromosome, and we are going to have this. So it is actually very good that we are going to have a genetic material which is altering it by using this particular virus at the genetic level in the germline. It means these viral vectors we are going to use for the somatic also and your germline also, depending upon our purpose. That's what we are going to use, the same concepts. Don't get confusion now. So we are going to have the same concepts which are used for the both the type of therapies. Now we are going to have a non-viral vector systems in the nucleic acid based delivery systems, non-viral. So one is a pure DNA construct. So this is in the gene therapy. We are correcting the genes by using a non-viral vector base. There it is not involving any virus. The first one in this particular case is pure DNA construct. So the direct introduction of the pure DNA construct into the target tissue, this is one. And the efficiency of the DNA uptake by the cells and expression, it is actually rather low because we are not to introduce everything, every cell into a DNA. This is actually what the problem in this particular non-viral vector, because we need to take care of every cell wherever it is got, it is need to introduce. But their success rates are very low. When the uptake is not there, DNA will not enter. When the DNA will not enter, genetic problem will not be cured. That is the reason, consequently, large quantities of the DNA have to be injected periodically to make entire thing into the implemented and effectively it is being used. This is actually what the problem of non-viral vectors using a pure DNA construct to introduce. Even though it is not intervention of any virus is not there, and the intervention is not there, viral intervention, but it is a long-term process and it is making it. Most of the times, if you see in the bone marrow treatment or anywhere, what they do, first they make entire body sterile by killing all the cancerous bone marrow cancer when it is present. The cancer cells which are present in the entire body, they will make it sterile, kill them. After that, they take the bone marrow cells of the either parent or a sister or a brother that is normal in nature without any cancer. They reintroduce them. So this intervention of making it in a sterile environment, killing entire cancerous cells of bone marrow and making them even single cell, it is not present. Again, it will be given a but. So we are going to give directly the bone marrow of there. There we are not using any vectors. There just it is a replacement therapy, transplantation therapy. Don't get confused with this gene therapy with the transplantation. In the transplantation, we don't have any intervention of taking a DNA and doing it. What we do, some patient of a bone marrow is present, the bone marrow got corrupted and it is keep on going, and you are not able to restrict it because the cells have got that heredity, that environment got changed, mutated. So in this case, what you are doing, we are killing entire body, whatever the bone marrow cells present in the patient's body in the controlled atmosphere and the environment, where we are using like a class 100 and the sterile. More than that, we need to use it in the patient because during the time, he will be, body is completely deprived of his defense mechanism. So even a small pathogenic organism, it will cause a lethal damage. So we are making it and we are giving without any change the bone marrow cells of the relatives, which is having a great matching, and we are introducing. Here we don't have any intervention. This is actually a therapy we are making as a called as a transplantation. transplantation, it is appearing as a pure DNA construct. What we have done here is the DNA of a normal we are giving directly to the patient, and it is replacing the original cells of the patient with the new cells which is not having any defect. Even though it is present, we don't call it as a gene therapy, we call it as a transplantation, that is bone marrow transplantation. We are doing it because that name why you are giving, we are not having any alterations in the gene, we don't have any alterations in the gene. So if you are making alterations in the original patient's gene and correcting them and reintroducing them, then we call it as a gene therapy. You need to make it a simple clarity which we need to understand now. Second is DNA molecular conjugates. So direct DNA constructs, we have already seen. The problem is we have got a entry is very less and we need to use huge quantities of DNA, that is actually making a lot of wastage. And DNA molecular conjugates, so commonly used synthetic conjugate is poly-L-lysine bound to the specific target cell receptor, and the therapeutic DNA is then made to combine with the conjugate to form the complex, and it avoids the lysosomal breakdown of the DNA. So the number of DNA will not be used, will be less and lesser, and it is becoming a more effective, because in the direct DNA construct, it is actually cleaved or entry and reaching to the cellular site is having a lot of barriers and problems, hurdles. So here we are having a conjugate. That conjugate, it will protect from the lysosomal breakdown of the DNA. So efficiency will be increased by making this particular modification. Here we are not using any virus. We are using a DNA with a complex. That is actually what you are having a conjugate with another moiety to protect its degradation. So we are going to use it is also a non-viral vector. The third one is lipoplexes, like your lipid DNA complexes are entrapping your DNA. Most of the times the liposomes are you are having taking a direct drug entrapped in the liposomal membrane, or the hydrophilic drug which is entrapped inside the liposome, or you may be having a tagging. So this is one is a liposomal one. In the lipoplex, what will happen? You are not going to take as a liposomal bilayer or a unilamellar and bilamellar. Even though if you are having a liposome, that lipoidal membrane has got a complex with the DNA, then we call it as a lipoplex. And we have already seen in the polyplex and lipoplex. In the polyplex and lipoplex, we are going to take a polymer, it is as a neosome or a liposome, and the polymer we are making a complex with DNA so that we are having a specificity. But the lipoplexes generally, if you see, if you don't have the liposomal membrane, if you are making it, it is good that it is having a flexibility and it is taking, we can take it. But the lipoplex is the lipid DNA complex. So the DNA construct surrounded by the artificial lipid layer, most of it gets degraded by the lysosomes, because the lipid layer is same as that of your biological membrane. So it is having accepted by the patient, there will not be any problem for the trigger release. But the exterior one which is having a lipophilic in nature, because the lipid complex which is having a lipophilic in nature. So the lipophilic in nature, it will make the engulfing of the material and cause the degradation, most of it by the lysosomes. Most of the gets it is degraded by the lysosomes which are present in the cell, and by the phagocytosis they will be taken and it will be lysed and making it a DNA not available. So the fourth one is human artificial chromosome. So this can carry a large chromosome with one or more therapeutic genes which we want to make it specific, and this will be used. Most of the times it will be such type of segment in the large DNA. And we are going some of the having a specific functions. So we can use that in the production systems also. Most of the times we are having a human specific enzymes we want to make it. In that particular case, the genetic material simulated exactly to the human being. In the previously what we used to take, we are going to take these materials of your hormones like insulin or any from the primitive animals by extracting their blood and concentrating them and taking that enzyme and giving it. There are a lot of side effects and reactions are present because of the this hormone which is rejection by the body because of the endothelial system. Both are different. So in the human artificial chromosome, we are having the simulated to the human DNA by making an alterations which we are going to have, and this entire chromosome is having alterations in the therapeutic genes which are having with one or more therapeutic genes which are getting having altered gene alterations, and which will be used for the specific purpose of synthesizing a defective gene or the defective area. Most of the times there are some approaches are present in the by doing all the above vectors, how to treat the gene therapy. We are going to enter and we will see that, then we are going to select a particular approach for our gene therapy. So how we are going to have that approach, which approach is going to be suitable for me, we will be knowing it only by knowing what are the approaches are available. So general approaches are a gene modification, gene transfer, and the gene transfer in the specific cell line. This is actually what the three approaches are there. So one is the gene modification. So the name itself, it is having it is a simple, it is a gene transfer and the gene transfer in the specific cell line. So in the gene modification, again we have got three types. One is gene replacement, gene correction, gene augmentation. This is actually what we are doing. The original defective gene is getting modified. And in the gene transfer, we have got again three approaches. One is a physical gene transfer, chemical, and biological. So this is actually three approaches. So gene transfer by means of a physical, and that is based on the size, surface, and all these things which we are going to consider physical parameters or factors. So that we are making into the use. The second one is a chemical, that is having a specificity with respect to enzyme, a receptor, or it may be a pH, or it may be a redox potential, like that. So it is another one is a biological. In the biological, which will be having a perfusion rate, or it may be having a specificity with respect to certain ligands. So that is having that biological, which is we are going to use them, these approaches to transfer it. So chemical mean, physical mean, and biological mean. Most of the times, this biological mean we are using the viral vector. We have seen in the gene transfer in the previous cases of the types. In the third category is gene transfer in the specific cell line. So in the specific cell line, somatic germline and germline. It means the approaches will be coming. The first, in that we have got the two approaches, and don't get confused, sir, we have already seen this chemical and defined. So if you are having entire thing into the account, so we are going to have a broader picture of where to classify it at what level and what way it is being asked. So what type of what are the different types of gene therapy, what are the different vectors of the gene therapy, what are the different approaches of gene therapy. If the question is specifying it, types or vectors or approaches, you need to write the classification based on the question what is being asked. So if you have been asked the gene therapy and classify it, then you can have this particular side is best. It is not mentioning anything because it is covering entire thing, and you are going to have in the biological treatment. So it is coming in the viral, and if you need a gene transfer, again you have got two vectors, that is viral vector and non-viral vectors. You can take it, or if it's somatic therapy, you can divide into the viral vectors and non-viral vectors. You can classify and link to that. So type of question, whether it is a types of vectors or the types of gene therapy or approaches of gene therapy, you need to write that particular answer in the exam. The first one is gene modification. In the gene modification, we are attacking at the molecular level. The gene is defective, it is not listening, or it is not producing desired effect. There are two things in this particular modification. One is, it is over-synthesizing it than required, under-synthesizing it than required, or not working well, it got silent, or it is got standstill. There are three types of defects which you are going to find: over, not at all, it is responding, or under. It is responding but under, not into the optimum level. So in the suboptimum level, which you have got either over or under, But in the case of present yes or no, that means whether the gene is actually functioning or not. It is a defective is completely shut down, or it is working with a higher and lower level, depending upon the defect modification of gene modification is required. Defective gene is modified to render it functionally normal. We are bringing to the normal. So where it is going to be applied in the defective gene, patient with a genetic disorder. So we need to take that patient where the specificity of the particular disorder. In this approach, we isolate the genomic DNA of the particular patient, and we are having a construction of the genomic libraries and matching with the original normal libraries and identification of the defective genes. So now what will happen? We are going to have a so many approaches are present now, the genomic sequences. Now human genome project it is taken a lot of so around throughout world has struggled now to have a human genome project. Lot of time, 10 years it took. Even our India has taken part in it. So thousands of dollars have been, that is lakhs of dollars, which have been used in this particular case of make it. But now it is a simple genome which can be made within a short span of time with very affordable price. We can make it now. So in this particular case, the patient genome is known, the sequence order is particularly known. And when it is mapping with the gene library of a normal human being, and when you are having it, most of the times in silico software itself, it will give the where the particular defect is present when compared to a normal. It is a that a defective gene will be known by applying this. And when that particular one, we are going to have a gene replacement. So the removal of the mutant gene sequence from the host genome, we find the particular defect. So we remove that, and it is replaced with a normal function gene. So it is actually what is called as a cut and paste. So we cut the specific gene and paste the correct gene. Now you may get the questions that it is appearing going beyond my head. How it will be possible? Like I'm cutting my paper and whatever the photo is present, I want to take the photo and pasting onto the photo which is not clear or blurred photo. I'm pasting on it. It is appearing like that. Is it possible at the molecular level? How it will be possible? Because even I don't see cell. In that I don't see nucleus. In that I don't know where the chromosome portion is defect, and how to select that one and cut it and paste. That is a bigger question you will be having. Most of the times you will be having little bit later when we are going in the depth. There is called as a CRISPR technology. We will be seeing that little bit later. So how this is happening and made it is a simple having a Cas9 enzyme and the CRISPR technology, both together they are doing wonders by making changes. We'll see little bit later, not now, what this CRISPR and how it is doing, how it is making cutting and paste, like as simple as that of what we are doing it, how it is possible precisely. So we'll see. I think if you go to internet, see who is Nobel Prize winners in 2020. If you go there, you will see that the scientists, so they have done the tremendous work in this particular same thing, made how the gene cut paste methodology. You can very well see it. If anybody is interested and finding a time, you can do it. So watch their videos and you can know in a little bit better. Of course, we will see those technologies a little bit later. So it is used for the disorder marked by the deficiency of the enzyme or the protein. So we are correcting it. This is actually what is called as a gene replacement. Most of the times a particular defect will be present, like you have got insulin is very less in the case of diabetes, and if the insulin is getting in the pancreas, so beta cells of pancreas, when you are having it corrected, then it will synthesize its own. Then if you are correcting at the genetic level, if it is getting treated, so we need not give this treatment regularly. So that is actually what the logic of correcting in the long term and genetic diseases if it is present. Another one is gene replacement. We are replacing it with a new gene which is already but corrected. The second one is gene correction. This involves the only defective portion of the mutant gene which is altered to provide the functional gene without precisely changing the gene backbone to its natural form. So it is we will not touch entire genome. We take only correct the portion where it is there. In the previous case, what we are doing, we are taking a removal of the mutant gene completely and replacing with the host genome, and we are making it. These are the T's. In this particular case, the particular portion which you are having it is being corrected. So the most of the times this functional gene which is having after correction, it will have a functioning correctly. Whether it is the correction may be by simple deletion. Sometimes the correction will be done by the deletion. So some extra fragment is present. The correction will be done by the simple deletion. Sometimes it will be insertion. So in the particular one which you are going to have a inserting particular component in that particular place. So most of the times, what way we are going to correct it, it is depending upon what type of the problem the patient is having. The third approach is the gene augmentation. Expression of the mutant gene in the defective gene is modified by introducing the genetic material and post-genomic before altering the defective one. So most of the times, this expression of the mutant gene is in the defective gene is modified. The mutant gene is modified, introducing the genetic material. So original genome which are having, so which you are going to have, so the original function is getting augmented by it is getting increased because it is got a mutation. So that is the